Hello, good morning, everyone, and welcome to today's webinar. First of all, I'd like to thank everyone for joining us today for the webinar, which is the Natural Products BioHUB Green Economy Center from Lab to Field, Biocontrols, Biologicals, and Sustainable Crop Protection. My name is Hamza Bhatti, and I'm the Research and Engagement Assistant with the Natural Products BioHUB Green Economy Center at Swansea University. And it's a pleasure for me to be your host and moderator for today's session. I can see there's quite a few people who've joined up and who've signed up for today's webinar. And it's a diverse audience today where people from the academia, industry, and the wider community have signed up for today's webinar. And I'd just like to share that today's webinar, it will bring together expertise across three organizations, which are playing an important role in advancing crop protection and biological solutions. I'm pleased to be joined today from our colleagues from CABI, Russell IPM, and Swansea University. Instead of me introducing my speakers ourselves, I'll invite each of our panelists to briefly introduce themselves before they begin their presentations. So everyone here who's attending can hear directly about their background and the work that they are leading. Just before we start, I'd like to share a few points with everyone that today's session, the purpose of this session is that it's going to be an interactive session. So I would highly encourage everyone to get involved. If you have any questions for our panelists during their presentations, please submit them using the Q&A function at the bottom of your screens. I'll aim to address each question after the presentation where the time allows. If we are unable to get your presentation, your questions during the end of the presentations, we'll go back and make sure to address them after the end of the session. Another thing I'd like to share is that after today's webinar, we'll also be sharing a follow-up invitation with everyone who's registered. The purpose of that session is to continue today's conversation in a more open and informal setting where it will give everyone the opportunity to exchange ideas, ask questions, and explore interest and discuss any potential opportunities. One of the things that we are really interested in is to explore whether there is a wider consensus in developing a Biocontrol focused community or cluster, which will bring together colleagues from academia, industry, and the wider community. So I hope today's webinar will be quite interactive and interesting for everyone. So with that, I'd like to invite our first speaker who will introduce themselves and begin the session. So over to you, Dr. Belinda Luke from CABI.
Thank you. I'll just share my screen. Can you just confirm that you can see that?
Yes, Belinda.
Perfect. Hi, good morning, good afternoon if you're listening from further abroad. So I'm Dr. Belinda Luke, and I'm in the biopesticides team in CABI. And I've been a biopesticide researcher for 30 years now, looking at naturally occurring fungus to control insect pests. And today I'm going to be talking about one of those fungi that we have found to control the cabbage stem flea beetle in the UK. So what's the problem with the cabbage stem flea beetle? Well, it was being controlled really well, and there was not an issue until the neonicotinoids were banned from use in the UK and across Europe as well. And since then, there's been a big cost to growers of damage through the insect. And you've got the larvae here that burrows inside the plant, but you also have the adults that attack the new leaves when they emerge just after planting. And in 2019, 2019, it was 79 million worth of damage. That damage now has actually gone down, but the damage has gone down because farmers are no longer producing oilseed rape due to the issues of the cabbage stem flea beetle. So what can we do about it? So initially, Cabby and what was then CHAP is now the UK ATC, went through the Cabby culture collection for UK fungal isolates, and we screened them to see which ones were suitable for killing different pests. And we found that a fungus, Isaria, was very effective at controlling the cabbage stem flea beetle. So the picture on the left-hand side is the spores that we've mass produced, and then the picture on the right-hand side, you see a white fluffy thing in the middle of the slide, and that is a cabbage stem flea beetle covered with the fungus. So we've done this work through a series of research project funding. So the first one we had was in collaboration with CHAP, or as they were then, CHAP, H&T, which is a seed coating company in New Zealand, and Russell Bio. And the idea of this project was we knew that we had this fungus, Isaria fumarosa, that it was suitable at killing the cabbage stem flea beetle. So we needed to know more about its efficacy and whether we could mass produce it. If you can't mass produce it, you can't turn it into a commercial product. So that was the idea before that. And there were four main outputs that we tried to cover through the project. So first one was end user engagement. It was important that we communicated this work to farmers to see what they thought about it, because it's a relatively new idea to them to put fungus on, very different from chemicals. So we wanted to bring them along the journey with us. We also looked at seed treatment development, foliar treatment development, and then mass production and scale up, and then handing over to the commercial partner, which is Russell Bio. So this is the life cycle of the cabbage stem flea beetle, and it goes through the various stages of adults, lays eggs, the eggs hatch, the larvae feed, the larvae pupate, and then the adults emerge. And within this life cycle, we were targeting two different stages. So if you look at the top on the left-hand side, when the adults were emerging in June and July, and also then when in September, when they were coming to attack the newly planted oilseed rape, we would try to attack the adults with a foliar application. Between July and September, so in August, the beetles have a hibernation period, so they're not around in August. So that's why we've got the two different stages there. And then with a seed treatment, we wanted to attack the larvae. So when the eggs hatch, the idea was that because the seeds have got a coating of fungus, the fungus will be in the soil, and then that would kill the larvae. So you would be attacking the cabbage stem flea beetle at two different points within its life cycle. So these are some results that we got from the first project. So this is looking at mortality over time. So at the bottom, we've got days post-infection, and then we've got percentage mortality on the y-axis. And we've got four treatments. So we have an untreated control. The insects were just put in a petri dish and nothing else was added. We have a control of tween because the spores were suspended in tween in our treatment ones. So we just had tween on its own, which was the black line. And then the two green lines are the fungus at two different concentrations. So we can see that the two controls, the black and the blue line, are very low levels after 16 days. And with the fungal spores, by day 14, you've got over 80%. And by day 16, you've got about 90% of the adult beetles were being killed. And very interestingly for this experiment is that even though the darker green line was half the constant, or approximately half the concentration of the light green line, it still worked the same. This is crucial because it means you can put on half the dose, which means half the cost of the end product. And I shall go in and talk about that a bit more where we further refined that. We then got a further project that was looked a bit more in detail on the formulations of the biopesticide. So this was a phase 2. Unfortunately, there was a bit of a gap because this work was affected by COVID. So there was a break in the research there, but we did manage to get a phase 2, which is just looking at formulations. And also what we also needed to do was just do a bit more work on the range of temperatures that the fungus grew up, because we're talking about applying in the summer when the temperatures would be relatively warm, but also we're talking about putting the fungus in the soil where the temperatures are going to be relatively low. So we did a range of temperature profiling so we could understand how quickly the fungus would grow in those environments, or if they could survive in low or high temperature environments. And then we also did further mass production studies to perfect that. So here we are. I said earlier that we then looked at more detail on the treatments of the fungus and tried to bring that down from the graph I've just shown you. So here we have the light blue line at the bottom is the control. We've got days post-infection along the x-axis and then mortality on the y-axis. And you can see here that the dark green line, which was a dose of 8 times 10 to the 7, so quite a bit lower than the previous experiment, where we again got good mortality above 90% by day 18, where the other concentrations do kill, but they're slightly lower, and we probably wouldn't go for those. Possibly we could do some more work on between treatment 2 and treatment 3, the top highest treatment, to see if we can still perfect this even more. And then the picture on the right-hand side shows a juvenile that has been killed by the fungus. So the fungus can kill the juvenile and it can kill the adult stages. So looking at the seed treatment, we have a series of pictures here, and starting on the left-hand side, we have a seed coating equipment. So you put your spores, fungal spores in there in liquid formulation, you put your seeds of the oilseed rape, and then it spins it around very quickly to coat the spores. We then took those coated seeds and put them in soil, which is the next picture, 2 pictures along. And then you can see that then we put the larvae of the oilseed rape in, and the larvae picked up the fungus. And in the middle picture, you have white dead larvae on top. The next picture to that, second from the right, shows fungus starting to grow out of the insect, and then the soil one on the far right is showing that. So that worked really well. However, we did have some high mortality with the seed coating machine at the higher temperatures, at the higher speed. Sorry. So again, we needed to do further work on that. And then we looked at survival of the fungus in different emulsifiers, which would be for the water-based application to control the adults. And we tried, well, here's data for a standard oil, which is in the green, and then 3 emulsifiers. And we were generally getting good viability, above 70% for most of them, until up to day 56. And then from day 70 onwards, it started to decrease. So by day 248, you can see that the blue treatment had very low survival of less than 20%. So that can knock out that emulsifier. So next steps, we have just recently won a DEFRA Adopt program in collaboration with Innovate UK. And this is where we hand over formulations to farmers, and the farmers will apply it on their oilseed rape. These calls are run by the farmers, so they are in charge of what they want to do. So those trials will be happening later in the year. And hopefully, if you can attend the new IPM symposium on the 8th to 10th of September, I can give you results then of what we found with these field trials. And on that, I'll leave it. Thank you very much.
Thank you, Belinda. So we can take a few questions. I can see there's a few questions that come through directly to me. Belinda, the first question that's come through is that what have been some of the biggest challenges in taking this technology from lab research towards the practical field application?
So I think at the moment, the challenge is in the formulation. And because what we try to do as much as possible is use the equipment that the bigger companies have already got. So for example, when I was talking about the seed coating machine, so it's not that we can't use that, but we need to build protocols to advise people who will be making the final formulations on, what speed do you set it at? How long do you set it for? I mean, things like that. So formulation have been the biggest challenge so far. However, we've not taken this to the field. And I'm sure when we go to the field in this next phase of the project, that we will also come against challenges. So it's things that we can do and we can adapt to it. It just takes a bit longer. But we know that the fungus is robust and we know that it works. So it's just perfecting the optimum of the formulation. And I'm sure there will be issues that come up in the field trials.
Thank you. So the next question is it possible to know the other fungi that were tested?
Well, there was a range of other fungi tested. I think we did it's about something like 50 different isolates. So they would mainly have been Metarhizium and Bavarius would have been the majority of the other fungi that we tested. Yeah.
And there's another question. Does this area of Fumarosia work on other insect pests, or there is no work done as yet?
We haven't looked at other pests yet for that. So no, I don't know that. But if someone wants to suggest pests that they're interested in controlling, then please let me know and we can look into that.
Okay. All right. And there's another last question that I can see that also. Just a reminder to everyone is that if you could just put your questions in the question and answer section, then everyone would be able to see those questions from the panelists. So instead of sending them separately. So Belinda, another question is that from those who may not be familiar with the cabbage stem flea beetle, could you explain why it has become a significant challenge for UK rapeseed growers in recently?
Yes. So the cabbage stem flea beetle has always been in the UK. Well, not always, but it's been in the UK for a very long time. And it was very effectively controlled by a seed treatment of a chemical pesticide, a neonicotinoid. That was banned by the EU. And since then, the only pesticides, really chemical pesticides, that have been left are pyrethroids, and resistance to the pyrethroids has increased hugely. So it's effectively left oilseed rape farmers with no control. And what happens is that when they plant the seeds, as soon as the seeds come up, the adults come and they eat the new seedlings. And quite a few farmers were finding that they had to completely replant. They would clear the entire field. And then if any plants survived that initial growing stage from the adults, then they would be attacked by the larvae. The larvae burrow inside the cabbage stem flea beetle, damaging the inside of it, and then it would break or it wouldn't grow very well. So yeah, big problem to farmers, and so big a problem that quite a few farmers now are not producing oilseed rape because it just wasn't profitable for them.
Okay, thank you for that. Okay, thank you, Belinda, for the interesting presentation and for sharing the work on the cabbage stem flea beetle. So now I'll just move on to our next speaker, just making sure of the time that we have. So if anyone else still wants to ask Belinda a question, please put your question in the Q&A section, and then we'll answer them right at the end of the presentation. So on to our next speaker is Dr. Naeem Hassan from Russell IPM. Dr. Naeem will be sharing insights for the industry perspective and particularly around biocontrol and practical IPM solutions. On to you, Dr. Naeem.
Thank you. Guys, can you see my screen?
Yes, that's fine. Dr. Naeem, if you could just close the presenter view.
Presenter view.
So right, the three dots next to the slide one, right at the bottom. Yeah, there's three dots in where it says slide 1 of 24.
This one?
There's three dots. If you just move it slightly up in the middle. If you just move your mouse a bit higher. Your mouse a bit higher. Yeah. So where it says slide 1 of 24.
Yeah.
Yeah. There's three dots over there by the camera.
Yeah.
Yeah. If you click on that one and just do end presenter view, and so we can see your full screen.
One second.
Yep, that's fine. Thank you.
Can you see that?
Yep.
Okay. Good morning, everybody, and good afternoon, wherever you are. My name is Dr. Naeem Hassan. I am at Russell Biosolution Teams. We have been working on biocontrol over 25 years and biorestoral solutions. So today I'm going to talk about biofertilizer and use in arable crops. And we have been working on biofertilizers. How can we increase we can grow arable crops, wheat, maize, and corn in a sustainable way. So Russell team developed a biofertilizer called Converter, and that is actually based on 3 microbes: nitrogen-fixing bacteria, Azotobacter crococum, and phosphorus and potash solubilizing bacteria, Bacillus megaterium phosphaticum, and potash mobilizing bacteria, Bacillus maceraceus, with collaboration with mycorrhiza. And then we tested this in different crops. Before going to there, I would like to give you a bit of information about the Azotobacter. Azotobacter is an important microbes, and it helps plant to utilize nitrogen. Nitrogen is very abundant in the atmosphere. About 78% of the nitrogen gas is available, but these are unavailable to plants. So plant has got a secret weapon, is the microbes, which breaks the diatomic nitrogen, which are 2 atoms of nitrogen bonded together that just far too stable plant to take it. So these bacteria produce a nitrosomonas enzyme, and this enzyme, nitrogenous enzyme, breaks these 3 strong triple bond, and these bonds breaks and they produce available form of ammonia and nitrate, which plant can take it. It's like they are working together with the plants. They break these bonds, and in return, plants help them to share their essential nutrient. These are very important. Nitrogen is an important source of potassium and nucleic acids, DNA, RNA, and without nitrogen, it would be difficult to life exist. So we look at how can we increase the nitrogen efficiencies on wheat in the UK. We have conducted a trial in Northamptonshire where the standard NPK regime was used, and with the NPK, how we used different dose of biological fertilizer converter in 50%, 70%, and 100% of with chemical fertilizer. Let's look at the results. These are the results of the yield, where 100% NPK and also different dose of converter in use. Converter use as a 6 liters per hectare, and we see about with the 50% less synthetic NPK, converter producing around 1.5 ton more yields. So it is a significant achievement. Then you can reduce the chemical fertilizer without compromising yield. And with chemical fertilizer, you get around 22% more yield. So this was a trial done in 2023, and we have conducted further trial in 24, 25, and we have a 3-year solid data. It's showing that biological fertilizer mixing with the synthetic NPK, whatever form we use, either urea, DAP, whatever form we use, it can reduce chemical fertilizers substantially and helps farmers to grow more sustainably and also reduce the cost when the turmoil in the market, when especially the fertilizer price is very high. And then in that case, it helps farmers to reduce their input cost. So in that mix, we have seen, yes, you can reduce the fertilizer, but what are the quality? The grain protein is an important factor. So we conducted further trials and introduced a new biostimulant with biofertilizer called AminoMax. AminoMax is nothing but a plant-based amino acids, which helps plant to build nitrogen profile. So we applied AminoMax and we applied Converter together. And this is the results. Then in this time, we applied Converter in 2 different doses, 6 liters per hectares and also 3 liters per hectares. In the first experiment was 2023, was high dose, 6 liters per hectare, and here we applied a lower dose, 3 liters per hectare. And the earlier slides I've shown you that we have an increase of 22%, and here is about an increase of 10%, 9%. But when we have applied Converter and our AminoMax together, we've seen the increase of the yield. And the most important thing is this slide. We see the amount of grain protein increase. Grain protein is an important factor for getting the milling spec right for the higher price. The higher protein content is important for bread making wheats, so the farmers get better price. So when we applied the AminoMax, we see about grain protein increase from 11.3 to 13.3%, 2% more proteins. So it is a significant achievement. Normally, our farmers, to get the protein content right, they apply 80 liters of urea twice during growth stage 69 and 71, flag leaf stage or before flag leaves. And this is 1 kilo of AminoMax actually is working to replace that 160 kilo of urea. So we also applied this mix, Converter AminoMax, in Yorkshire, in our north of England, and where we have a farmer who said, if this biofertilizer, it works in my field, then I would believe that it would be working anywhere. And if you look at this patch of land where fertility is very, very low, normally it's got not a good history of at high yield or get a good yield from there. So we applied Converter, and this is a untreated control and the Converter treated area, and there is a significant yield difference, about 1.4 tons more yield. This is a good achievement of these biologicals. It can show that biological can reduce the yield, reduce the chemical fertilizer and increase the yield. So therefore, we took it further up and we take it to the Europe. So whether it has got any effect on any difference in different locations. We have conducted a trial in Spain on barley in different doses of Converter and AminoMax, 6 liter Converter, 2 kg of AminoMax, 3 liter Converter, and 1 kg of AminoMax per hectare. And there is a significant difference of yield while we're using the 6 liter of Converter and 3 liters of converter. So the both cases is give a significant difference from the untreated control and was following the same trend. Then we further applied converter in sweet corn in Greece by using, and also compare with other standard in the market. Look at the results. The converter in two applications and 1 applications. Normally we have applications in the both cases, yield comparisons and the moisture content is a significant difference from the other standard in the market, biologicals and chemicals. Now look at the converter is giving us the good results in herbal crops. We had conducted some trials in our field crops carrot, and we have seen when you apply the converter and growers use a half dose of calcium nitrate, and there is a significant difference in the 100 carrot weight. So it's showing that this has got potential in field crops, edible crops, cereals, and as well as the vegetables. Further trial has been conducted the 2024 on sweet corn in the UK and at Sussex. The farmers, we introduced a few biostimulant that we've noticed the sweet corn growers, when temperature drops during the period of September, October times, corn filling was a challenge. And the corn is half filled, it doesn't grow fully. So by bringing the secondary crop formation, we call it, and the converter with 2 other biostimulant, Yokosan. Yokosan is an optimized blend of humic acid, amino acid, and seaweed extract, and Recharge is a microbial biostimulant. Both together is applied with a converter, and we managed to get a 80% over control the crop formation. And so this is a real breakthrough for the growers, that if you use the biostimulant, it can support the plant physiology and bring the highest potential and can deliver the crop completely filled, and then they can access the benefit of the biostimulant. So that's all we have. So far I talk about biofertilizer and impact on different crops. And this year we have been introduced a new novel biofungicide based on Pythium oligandrum. This product has been registered in the UK, and we are distributing these products. And this Pythium oligandrum M1 strain actually works by 2 modes of action: mycoparasitism. It's a beneficial fungus grown on a parasitic fungus, and also it supports production of growth phytohormones. So it's been listed against wheat diseases and oilseed rapes, especially Fusarium, Septoria, Pythium, Rhizoctonia. We have data on conducting trials on different growth stages, growth stage 36 and 39, 1 application, and another application is 67 and 70. The application rate was 0.1 to 0.2 liters per hectare or 0.1 kg per hectare, 2 formulation, OD and vegetable powder formulation. And if you look at the damage on leaf on y-axis and the various dose of polyversum, and the polyversum different doses and various formulations, OD and WP, is compared with chemical Plaxo and Serenade and other biologicals, and it showed the similar trend of damage on leaf percentage. And also we look at the yield percentage, and the polyversum in both cases, OD and WP, with 2 different doses, 0.1 and 0.2, significantly increase the yield as well. So this year we're conducting a series of trials in the UK with the farmers and also with AHDB to conduct to see whether polyversum can have an effect. Especially there is a lot of interest from wheat growers on Septoria. Septoria control, we don't have much of a choice. The chemicals are available, is very limited. And so the growers wants to have a Septoria control. So we are conducting trials with the AHDB, and it will be it will be the results will be coming in future, and we'll publish in due course. So with this, I would like to thank you everybody for listening our talk, and if you have any questions, please feel free and ask. Thank you.
Hamza, you're on mute.
We can't hear you.
Thank you. Sorry about that. Thank you, Dr. Hassan, for the presentation. So I'll go through a few questions that have come through. The first one was, can we test this in pink bollworm? I don't know which product they were asking about.
I think it's mostly related to Belinda's talk, maybe not
Belinda's
mine.
talk, yeah, that's what I thought. Yeah, come afterwards. Okay, so yeah, we'll leave that for Belinda to answer. So the next one is that why did converter and AminoMac increase the protein content, while converter ×2 and AminoMax did not produce a similar result?
You see, this is a very interesting question. When converter is targeted to reduce your chemical NPK and increase the yield, we apply one applications in the beginning, or when the plant is either growth stage 39, early growth stages. So we've seen 3 years trial. It increases the yield, and it can reduce the chemical fertilizer. But when it comes to protein content, converter is not applied on that time. So protein content is the latest stage, flag leaf stage, growth is 69 and 70. That time, our growers, they apply foliar urea on the time of the time of flowering. So it goes inside the flower and it helps to increase that protein content. So therefore, we introduced AminoMax. AminoMax is natural amino acids come from the soybean. Okay, it mostly contain proline and glycine. So these 1 kilo actually replacing some field as 80 kilo urea or 160 kilo ureas. So with the here addition of AminoMax is supporting a biological mix, that the biofertilizer helping to reduce the chemical fertilizer, and the milling spec, which is allow farmer to get higher price. I think for our listeners, there's a 3 category of wheat we have. One, the higher the milling spec, this goes for bread making. The second grade goes for biscuits making. The last grade goes for animal feed. Okay, so the higher the protein content, the better price they get. So to increase the protein content, we add the AminoMax with it.
Okay, cute. So the next one is, from your point of view, which kind of formulation is better to improve the EPF virulence and suitable for field application? Okay,
I think it's maybe the talk about
that's why I just that's again for Belinda.
Or maybe also could be related to our polyversum. We have seen in polyversum both formulation works very well. Polyversum is a Pythium oligandrum. It's OD and WP. Currently we are working on WP, and WP water solubility is good, and it can easily penetrate to the leaf surface. So if we soil applications, it can easily spread. So I think the OD is developed more onto the foliar, and WP is developed more onto the soil.
Okay, thank you. Okay, so would the converter differ in efficacy on different weather conditions? For example, summer versus winter planting?
Well, that's what we tried. That's a very good question. We tried in different locations trials. We tried in the UK, where we have the temperature is low, and we did in Greece, where the temperature is moderately higher. And also we did a trial of converter in Indonesia in rice. So we are across the crop and across the region, we found that there is no difference on the activity of these microbes. But bear in mind, microbe does not work below 10 degrees, and microbes does not work over 35 degrees. So when it goes over 35 degrees, it does not kill it. It becomes dormant. When temperature drops, it activates itself and come back again. So for our trials, we did not see any differences. We see the results are consistently good, and it can reduce chemical fertilizer and increase yield.
Okay, thank you. The next question, is the polyvasum soil-borne, or we have to apply it during each growing season?
Well, any microbes, including polyversum, we have to apply. There will be some sort of loadings will develop in the soil, and this is collecting from the soil. The number is very low right now, so therefore we are feeding into the soil and the crop. But if we apply constantly few years, we will have a reserve in our soil. But at this stage, we will say apply every year. But in future, it will have a sustainable growth, and we will have to use less, not as the dose we are using today.
Okay. So the next question is 2 parts. Can converter bio-NPK partially replace chemical fertilizers? If so, by what percentage without compromising yield? And also, how close do you think we are seeing this to become common practice on farms?
Well, very, very good question. Again, if I go to the first one, our result, we have found 50% fertilizer can be reduced, but it is sometimes people say you are very radical. 50%, I don't want to take risk. But as a safe side, 25% is very safer option. You can reduce 25%, 75% fertilizer, chemical fertilizer, 20, and the converter can without compromising yield. But if somebody is brave enough want to take the lead, then in so far our 3 years solid data showing you can reduce 50%. But the biologicals, our farmers in the UK still they found biological is a bit challenging. It's not like many people doesn't want to move their chemistry to the biologicals because it's working for me. Why should I move? It's a new things. Everybody's scared of the new. So new changing systems, and it's not only that only the biofertilizer changing, it's the changing of their spray regimes, it's changing other crops, is it comfortable to this, comfortable with that. So therefore there is a hesitancy we see on adopting biologicals. Okay. However, we have this year we have a very good interest from our farmers. We're working with also some farming cooperatives in the UK, and they buy together, and we see adoption is coming through, but it's slow. However, in Europe, it's far more advanced. And also we work internationally in Asia and Africa. We see the adoption, the acceptance is far higher. Okay, but we are catching up in the UK slowly and gradually it's coming in.
Okay, I think we have some time, so we can ask a few more questions, and then we'll go over to Vanessa. So while selecting the trial plots, did we consider the plots that are never exposed to conventional ways, including fertilizers, pesticides, or is it vice versa? And if it's the opposite case, the soil that is used to conventional fertilizers, will it be able to adapt to organic fertilizer and bioorganic stimulants? Quite a long question.
Okay, first of all, yes, these are quite compatible. We have done a thoroughly a reverse compatibility test. These microbes are the bacteria, so they are the nitrogen and phosphorus and potassium, whatever we're using, they are compatible. It can be used traditional fields or a field where less fertilizer been used, less fertility, it can be used there. But important thing is the temperature regimes. We have to make sure it's not too extreme, like either too low or too high temperature, and it is compatible. And second question was,
if it's the opposite case, the soil that is used for conventional fertilizers, will it be able to adapt to organic fertilizers?
It is also yes. In soil we have to first build the soil profile. We need soil organic carbon and soil organic matters in the soil. So if the soil is very like a less fertile, a less organic matter. So we need to focus on increasing soil organic matters. And both scenarios. Scenario number one, you have a chemical fertilizer. Scenario number two, you have organic fertilizer. They are compatible to both sources. Normally other biostimulant which is commonly used, seaweed, a biostimulant like amino acids, biostimulant like humic acids, converter is compatible with all of them. So I think in a safe side, we can say, yes, converter can be used, both chemical and biologicals. However, if you are concerned, use a small area, always a tramline, then gradually can increase your area of but one thing we see, the farmers wants to see better yield, but with the converter we never see the yield compromise. Your yield is always higher, either you reduce your fertilizer or not. If someone says, but in the UK we have mostly this has happened, I don't want to cut my fertilizer, I want to see the increase. So if you add converter with the conventional chemical fertilizer, you get X amount of increase. And we see over the maths stacks very well. We see over 1.5 tons yield increase. And converter price, if we take out from the yield increase, it makes sense that we should introduce biological fertilizers.
Okay. Thank you, Dr. Naeem. There are a few more questions, but I think we'll go on to our next panelist, and then we'll come back to these questions at the end of the presentations, so we can have another open discussion on that. So over to our next panelist, Dr. Vanessa. Thank you.
Can you see my slides?
Yes, yes, Vanessa.
Okay. Thank you, Hamza, and thank you for the brilliant panelists and for their excellent presentations. Thank you for everyone who is joining this webinar organized by the Natural Product BioHUB and focused on biocontrol agents and how to take them from the field to, from the lab to the field. My name is Dr. Vanessa Mirikash. I work with the BioHUB. Hamza is my colleague, and today I'm going to speak about a certain technique that helps us take these biocontrol agents from our lab, where we isolated them, to our field, where we can apply them to promote plant health. This technique is whole genome sequencing. Today we will try to explain how it can help shape the future of biocontrol. As most of you probably know, this technique is increasingly recommended by EFSA and a similar organization for any registration of bioproducts. Actually, for microbes that are used directly within the food chain, it is required. And for the plant protection microbes, it is preferred and strongly recommended in many cases. So to explain the whole genome sequencing, we will start with the first question, which is, what is the genome? We know that the DNA is the chemical molecule that stores genetic information, and that the genes are segments of DNA that can build proteins. And the genome is actually the complete set of the genetic material that is included within an organism, or in our case, a microorganism. So if we think of it as a book, we can say that the DNA is the alphabet and the genes are the sentences with meanings, because they are translated into proteins. And then the genome is the whole book of instruction on how our microorganism can function. So in a nutshell, the whole genome sequencing is simply a laboratory technique that allows us to determine the whole genetic material of a certain organism. It goes from the microbial sample to the extraction of its DNA, and then to determining all the sequences, obtaining the sequencing data, and working on bioinformatic analysis in order to give meaning to this data. Therefore, it is clear that whole genome sequencing is a powerful tool that helps us understand our microorganisms, and it supports the registration process. Now, in order to explain how it supports the registration process, let's first explain what the registration process is. It's going to take longer than this presentation to do that, but I'm going to give a short summary. And the registration basically goes from the discovery of the agent, its characterization. We must test it as as Dr. Naim was explaining, and Dr. Belinda as well. We have to test it against the target organisms and then take the test into the fields. We have to assess the safety, if there is anything dangerous that can be released by this microorganism, and then prepare the dossier and, of course, commercialize our microorganism. So it's quite a long process. And let's see how genome sequencing can help us with that. Of course, the first thought that we can get is that it helps with the characterization of the microorganisms. And that is true, because the whole genome sequencing can give us high-quality genomic data after going through the assembly, which is the reconstruction of the genome, and the annotation, which is the understanding of the functions of the gene. We can obtain high-quality data that will allow us to compare with the availability on the database and to explain exactly what is the organism, what is its molecular taxonomy, and where it sits within the classification. However, the whole genome sequencing takes us beyond the concept of a species. It can also explain what a strain is, because for a certain species, each strain can behave in a different way compared to the other strain. And the whole genome sequences can also can spot these differences between strains and maybe translate why, for example, a certain metabolite is produced by a strain but not by the other. Therefore, we can say that whole genome sequencing helps us understand or answer the question of what is the microorganism that we have. But we can go further with the whole genome sequencing to understand what are the functional genes in our microorganisms, so which genes are actually associated with beneficial traits that are important for us, such as, for example, when it comes to metathesion, endophytism, and entomopathogenicity, two of the most important traits that help us with plant health. And we can also, based on that, understand the mechanism of action of our microorganism, and obviously to predict how it's going to help us promote plant health. And if we understand the mechanism of action, we can conclude the most suitable application of the microorganism, and this will lead us to a better understanding to the product we are developing and to improve field performances, because if there is a certain way in which our microorganism functions, we can adapt our plant protection plan depending on that. So it will give us data on the mode of action and the best application. And of course, it helps us answer another question, which is how does the microorganism actually work? If we dive a little bit deeper in that, we can also speak about the biosynthetic gene clusters. This is very important when you have whole genome sequencing data, because it will help us understand which natural products are actually produced by the microorganisms and released. And in this case, we will understand very well the genetic potential of our microbe, and also explain its biocontrol capabilities. It guides us towards improving our strain, and of course, to explore which compounds can be produced. It could be, of course, novel compounds. An example of that is simply the compounds that biocontrol agents use to suppress other organisms, such as antibiotics, antifungal compounds, for example, ones produced by Trichoderma, insecticidal compounds such as destructin produced by Metathesion, and basically any other secondary metabolites that can be used within the chain of production of plants. However, if we're talking about natural products, we cannot ignore the fact that natural products can also be toxic. So the whole genome sequencing helps us reveal the presence of gene clusters that can lead to the production of toxic metabolites. And therefore, it can help us address certain safety concerns, which, as I understand, several of you are within industry, and this is a very challenging part of registering a certain product. With the whole genome sequencing, we can assess several parameters, but maybe we can focus on two. The toxic metabolites that can be released within the environment, such as mycotoxins produced by, for example, trichoderma, a very famous biocontrol agent. And with the whole genome sequencing, we can be directed into testing that, into assessing the quantities, in which conditions it can be released, and therefore it can guide us towards selecting the best species, maybe the strains, maybe the ones that don't produce, or maybe the ones that are that has a low potential to produce. And a very important part of understanding how genome sequencing can help with registration is by understanding the antimicrobial resistance genes. Because for this specifically, EFSA highly recommends to do a whole genome sequencing before registering a product, because through this technique, we can understand if our agent acquired antimicrobial resistant genes, if there are any families within its genome, and of course, if it has the potential to transfer these genes to other microbes. This shows that the genome sequencing helps us demonstrate if our microorganism does not pose any risks on human health, on animals, and on, of course, the environment, as well as other beneficial non-target organisms. So the whole genome sequencing can give us data on the safety in general, and it helps us answer another question, which is our microorganism that we are releasing in the environment safe? Another important data that the whole genome sequencing can support is the stability of the agent, because if we analyze several batches, we can end up with comparative genomics, and to understand if this strain is actually stable once we grow it in different conditions. We can understand if it acquires certain genes, if there are many mutations happening, if it's going to lose its virulence, if it's going to stay effective or not. And we can confirm the genetic stability of our strain, and therefore we can end up with more documentation to support our registration, but also the commercialization, because no company will want to commercialize a non-stable microorganism. So we can obtain data about that as well, and it means that whole genome sequencing also helps us to answer the question, is the microorganism stable? So to conclude, we can go back to our slide, which explains where does the whole genome sequencing sits within the registration process, and how it can actually support it. We can say that it supports it in each step. In discovery, because it can lead us to the selection of the best strains. In the characterization, because it can give us a specific identification and taxonomic placement of our strain. In the efficacy testing, because we can understand several points within the mechanism of action, and we can understand how to test it, and in which which are the best environment in which our agent can function well. And of course, the safety assessment is a big part of the whole technique, because it can allow us to detect any toxin genes or any microbial resistant genes. And of course, it can help us to understand the stability of the agent and the potential to commercialize it. So at the end of the day, we will end up with more evidence to strengthen the dossier. Therefore, we can see that whole genome sequencing does not only help with the characterization, but it can help with every step towards the way in order to obtain a good and strong registration dossier. So I just wanted to say that the Natural Product BioHUB sits on expertise when it comes to genome sequencing, and we will be happy to collaborate with anyone who would like to enter this interesting topic. And thank you all for listening.
Thank you, Vanessa. Okay, so there are a few questions. The first one is, from your experience working with industry, what are the biggest challenges companies face when incorporating whole genome sequences into the product development and registration program?
Well, the company, I think that the biggest challenge would be to understand how to annotate, how to reconstruct the genome. So I would say that this is the biggest challenge if the company does not have an expert working on that, which is not the usual case. Therefore, for me, to do that, the best thing is to have collaborations with academics who have the expertise to work on that in order to take care of this part of understanding the genome and how it can be used to understand the microorganism.
Okay, thank you, Lisa. And the next one is that how early in the product development process would you recommend beginning the genomic data for registration dossier? Is it something that should be considered straight away or during the development of the product?
Okay, so I would not do it at the beginning, as it can be an expensive and a long process that may lead to waste in a way. So I would do it only on strains that I've tested, at least preliminary tested, and obtained good results with. Those are the strains that would be interesting for a commercialization. Therefore, those are the strains that I would recommend to test. So not at the beginning, obviously not at the end, probably when we start getting some good efficacy results.
Thank you. And there are two more questions. When you said the first one is that for smaller companies developing microbial products, what would you say are the most common mistakes when they are preparing a registration dossier?
Okay. While I think that the best person to answer this question would be Dr.
Name, yeah, I was going to go there.
Yes,
Yeah.
exactly. I can only answer it within
Exactly.
A molecular biology point of view, and I would say that it would be the lack of expertise. That's why applying for projects with academics would be, you would get a grant, and it would be the best way in order to understand these different tests rather than the company doing it itself. However, I can only answer it from my point of view.
Fine.
Probably someone else with that experience.
I can help if it is okay.
Yeah.
What it is, if you're developing a dossier, we first need to consider that whether the microorganism we are target organism is effective of a wide range of host. For example, if I am working on Escherichia, I don't have only Cabestem flea beetle. I would like to have to other pest, whitefly. I have to see the aphids. I have to see the thrips, how it behaves. Commercially, when one single organism killing a number of pathogens or pests, then it is beneficial for me. Number one. Number two, is it high spore producing? Is production is easy and cost effective? If it is really difficult to produce, because recently we have our experience, we have a Metarhizium from Africa, which we bring it in, is very effective against fall armyworm, but is a nightmare to grow. It takes a long time. There is no growth until 14 days, 15 days. So that's no good for you. And number three is it stable in formulation? If it is stable in the formulation, then yes. With this tick, then we can look into the sequencing the gene. If it is not early, this checkout is not done properly, then dossier will be a waste of money. So we need to have first wide host range, high spore producing, stability. Then
yes, that is exactly why I said that the genome sequencing would be done after we get some good robust data on efficacy rather than just doing it really at the beginning. Of course, for us academics, it would be lovely to do it from the first day.
yeah.
Okay, and I think the last part of the other question that was part of this one, I think that is also answered by this one, is that what distinguishes a strong microbial registration dossier from the one that is likely to face delays during regulatory review? So I think that's probably answered with what you guys.
I can emphasize on the fact that we do have certain guidelines when it comes to the test that we are applying, and we do have certain recommendation. And I do believe that a strong dossier should consider both, not just what you have to do, but also what you are recommended to do. And a good and robust data on safety would be very, very important, because the efficacy has to be there from the beginning, but the collateral effect must be very well studied before just releasing a microorganism within the environment.
Can I add one thing? A good dossier has to show that this microorganism is free from contamination. There is no contamination there, and it is within the range of the secondary metabolites. Okay, the metabolites is produced. There is a range which fit on the range. It is free from all contamination, free from all entomobacters. Then yes, it will be looked at, and also it got enough robust data to support the dossiers. The dossier is completely full, and there is no question. But if when there is a weak dossier and they can put questions, then it will cost more.
Okay. Thank you, Dr. Naeem, for that. I'm just conscious of the time right now, so I'm just going to open the floor now and go back to some of the questions that we had from to our panelists. And also, if there's any questions that someone would like to ask directly, please just raise your hand and I'll come to you and you can ask the questions yourself. So some of the questions, I think, Belinda, Naeem, and Vanessa, if it's to anyone, please just answer who it's targeted for, please. So the first one is, are there side effects to organisms and other water plants in rice farms grown in wetlands? So I think that was for, if Wanja, if you could just retype that question again, and then we can answer that as one. So another question was the biological control of fungus using trichoderma. Our problem stays in making formulation. How did you do that at your level? So that's for Dr. Naeem and Belinda, if both of you would like to answer that one.
After you, Naeem.
Okay. Well, the trichoderma production, mass production, is been in the market long time. That is the most important thing is you have a clean production. That you don't have the contamination of aspergillus, you don't have the contamination of the other funguses, and therefore I think it's very important that the production area handles only one organisms. It does not handle multiple organisms. If you handle one organism and you have the right safety measure in place, the cleanliness and the lab protocols are followed, then yes, you will have a good formulation. But if you are same commercial setup is using for one day trichoderma, next day matresium, then another week some bacteria, you're bound to have some contamination, and especially clean air. Clean air is the key. We need a clean air, clean system where the microorganisms are not actually visible, seen to us. They are on the atmosphere, they are on our breath, they are on everywhere. So it got to be a clean facilities and dedicated team. Then I think this can be ruled out and you can have a good formulation.
And then just to, yeah, that's all good. But just to add to that, when you're looking at making formulations, you need to look at what the farmer has. So if the farmer doesn't have a sprayer, then you don't want to make a formulation that's has to be sprayed. You need to then think about granules that the farmers can spread onto the field. So you really need to look at, for the formulations, what the end user is going to use. Is it going to be a liquid formulation that's sprayed? Is it going to be a formulation that needs to be put in the soil? So there's lots of different things, but that's where we always start from. What is the farmer using currently that we can then make his life easy or her life easy by giving them a formulation that will fit that need. Thank you.
The next question is, I want to know if the use of bio-NPK doesn't have any residual toxins after prolonged use. And I want to know if it can be used instead of usual chemical fertilizers that we use.
So maybe for me, bio-NPK, so far, the literature and the published paper, there is no negative effect. Okay. However, microorganism doesn't mean it could the amount of loadings we are using in our formulation is very low. Okay, so it should not have some negative impact on environment or toxicity on plants, because these are from the soil where they are coexistence with the plants. They are from the atmosphere, and we just taking them, spread them, increase their numbers, putting into a formulation, bringing it back to the and in the formulation, the material we use dextrose, which is a kind of a sugar, and also we use sometimes talcum powder. So these are the lignites, which are natural products. So the impact is so far not reported any negative impact on plants.
Yeah. Thank you for that. There were two more questions for your presentation, which have come up, and I'll just go through them, and then we can then conclude today's webinar. The first one was mentioned as that you mentioned that in the trial by Converter, you used only 1 liter per hectare, and whereas the recommended liter was 3 liters. So why was that? And do you see that there will be higher benefits if you use the recommended dose?
We use the various, this product, we use the various dose, 3 liters, 1 liters, because when we use 3 liters we are using, then our farmer saying it's too expensive to us. Okay, can you reduce the number? Can it be possible? Because the reluctance of the hesitancy from the farmer was they don't want to increase their cost. We have to show that when you add the product with your current regime, you can get X amount of more yield that will financially viable for you. You can spend 36 pound, you can get 300 pound. We can show that. So they still farmers saying, why should a technology I don't know, I don't want to. So therefore we look at can we use the low dose, and the low dose is still we see 10-11% more increase, and the higher dose we get 20% increase. So the going future, what we are trying to do to get the loadings of microbes, which is in 3 liters, can bring into the 1 liters, make it more concentrated. Okay, therefore I know this may be confusing for a few people, so we use it. Why we're using different dose? 3 liters is a recommended dose. Why using lower dose? But we want to show that in low dose it works, and also it gives the edge which farmers are looking for. Once farmers is convinced, he can go to the higher dose.
Thank you. The last two questions. The first one was that in your 2024 results, where you compared Converter and AminoMax, it showed that you had 13% grain protein milling when you used 25% less NPK and no foliar. So how significant do you think the results are for growers who are supplying to the premium milling markets?
It's a very significant data, because I did, because time constraints, I did not show the economics of it. If you compare giving us a bit of an idea, 80 kilo times twice, 160 kilo of urea using a foliar, and a ton of urea is about hitting now 600 pound. So it's a substantial sum, and our is only 1 kilo of amino acids. So which is actually why the difference is because the assimilation, that this amino acid is assimilation and absorbed by the plant is high rates, and is a natural products. And the chemical urea, there is a big loss there. Okay, so therefore I think this is a very the milling spec achievement wise, we we got to 2% more protein with using 1 kilo of AminoMax with it. But if we don't bring it in, then the farmers see the benefit yield increase, but they don't see the milling spec. They don't see the protein content increase. So therefore, though he's adding a little bit of a cost more with their inputs, but adding the kilo of AminoMax, it can reduce, it can benefit them two ways. One is amount of the spray they do, and the second is the cost and handling of and storage of the urea for the later stage of the plants. So I think it is a very, very good. And recently we have a cereal exhibition in Diddley Scott Farms in Claxton Farms, and we saw a lot of interest from our growers in the UK moving towards AminoMax.
Thank you, Dr. Naeem, for that. There's a question for Belinda. Where did that go? The question was, how do we break farm? Oh, I think you've answered.
Oh, sorry. Shall I answer again? Sorry, I've typed it in.
Fine. How do we break just for everyone? How do we break farmers' denial to use biocontrol agents? Farmers are reluctant to use these products despite being effective and safe.
Yeah.
Good
So,
question. Yeah.
Thank you. So I think part of it is awareness. Not all farmers really understand. Well, some farmers have no awareness of biocontrol, and then some, they've heard the word biocontrol, but what does it mean? And it's quite confusing. You've got fungi, you've got bacteria, you've got biostimulants, there's a whole nematodes, there's a whole range of different biocontrol agents. And of course, fungi, they may have been in contact with fungi because it's a pest on their farm, it's a problem. Wheat blast or something, and they're like, oh, we don't want to put a fungus on, that's bad. So it's awareness is a big thing, and that's why we try to work with farmers early on and engage them and work through them. So that'll be one thing. Second thing is product availability. So we work with farmers early on, but the downside of that is that the product's not available. It's not registered. It's a research program, and they're going to have to wait 8 years before the product, 8 to 10 years before the product's available. That's too long. They've lost interest. So that's a big issue. When products are registered, are they available in that area? Can the farmer get hold of them? So if the farmer's in Scotland, can the farmer get hold of it if the farmer's in Cornwall? And that will partly depend on the distribution of the product. So you're relying on a third party there. And then another issue is cost. So as Naeem was saying, sometimes they cost more if you're comparing shelf prices, and it's not clear to the farmers, and why should it be, that actually when you use it, you're going to get more yield. So then you make more profit. But that's very hard to see in a comparison of shelf price. So that's where we need to do the work that Russell is doing, where they're doing field trials, they're showing the yield, they're showing how much profit is being made, as opposed to what's the cost of the initial product. So I think a combination of those things.
If I may add something to that, I believe that also the regulations play an important role in that, because the reason why we are shifting towards an IPM production system or an organic production system is because of the regulations that are now requiring that. Most supermarket chains would not sign any contract with the farmers unless they have specific plans to reduce the pesticides depending on the supermarket's requirement. And obviously in the UK and in Europe, the regulations are moving towards decreasing the use of pesticides. So I think that's an important factor that plays a role into convincing farmers to shift towards more sustainable production systems. And we all know that also when it comes to awareness, the customers are also more aware now of the side effects of pesticides, and that and it's change at a low level that can trigger change at a higher level, which is at a regulation level, let's say. So just adding one factor to the list that Belinda just said.
I would like to add a line with the Belinda's things. Actually, why they change. Farmers will take biologicals. They have chemicals. They are used to it year on year and year. They're seeing the same results, and they are busy people. Farmers are very busy. They don't have the time to read of the biologicals, and biologicals got is extra precaution. You cannot mix with that. You cannot mix with this. You can have to use it in this time. You can have keep it in this storage condition. They said, what's this damn I have to do? I have to do something clean in my head. I want to do the things which is I've been doing for last 10 years. They will only do when they have to.
Yes.
And otherwise, and this environmental benefit and this, and only when it hits their pocket. Okay, they will, this year, when the Middle East crisis began, we have a news that there will be billions of meal will not be produced because of the ERA cannot produce the fertilizer. Okay, so this is, but biological, I think we all need to handhold the farmers, increase awareness, and also need less regulation. Too much of a regulation. 8 years of a pesticide being available to farmers, they can't have it. And also, we would like to, our IBMA and everybody's working together, that how can it easily accessible to the farmers, and need more policy level discussion, policy level dialogues, that it makes the policy easier them to adopt, and they can take, and the efficacy they see is consistent. The many biological products in the market are not as consistent, and consistency is not there. So we have to have a product which has got consistently performing, and also the needs training of the farmers when to use it. Biological products often works when they use from the beginning, but beginning nobody wants to apply the product when there is no problem. Like this is one big things. Like when our kids, we give them better food, then they become nourished, they get healthy, they don't encounter disease later on. So in plant, we have to change the mindset. If I look after my plants from the beginning, I put enough nutrient, I put enough biologicals in the field, that later stage I don't see the surge. If that mentality comes in, it will be easier. All not dull and gloom. There are some hope. We see there's some, there is within few days, there is a regenerative agriculture, regen agriculture exhibition in the UK. Okay, there are farmers are paying £350 to come in to the exhibition to see the biological practices. So groundswell, you can look at it. Some farmers are coming, they're listening what's happening, how can we farm ecologically. So it is happening slow, but we need to keep pushing, keep going, doing the same thing, then it will go there.
Yeah, just conscious of the time over there. Thank you for that one. And just to add to all the three panelists, what you just mentioned about the different registration problems and everything, what I'd like to say to everyone here is that I'm just attached a document for everyone to have a look at. If anyone would be interested to know more about what we've spoken today with our speakers, we're organizing a international symposium at Swansea University with Cabby and Sams and Russell IPM as one of our main sponsors. So if today's webinar is interest to anyone who's attended today, and attending the symposium would be very relevant, as the symposium normally brings around researchers, industry, and growers, and especially will address some of the questions that Naeem and Belinda and Vanessa just mentioned about the registration and microbials. So please do register for the symposium and have a look at the flyer. And if you have any questions, please reach out to the email address over there. I'm just conscious of the time right now, so I'll just like to bring this webinar to conclusion. First of all, I don't think we have any more questions at the moment. So just to thank all of our panelists today, Belinda, Naeem, and Vanessa. So thank you very much for your excellent presentations and for sharing the valuable insights with everyone who attended today. We had a really, really good interest and number of people who've attended today. And it's been really interesting to hear the different perspectives of industry and our colleagues, especially from a company from Russell and Belinda with Cabby and Swansea University, who where we've covered quite a lot of areas of research in biological control. So once again, thank you for everyone who's was able to attend today and asked all the questions. We really hope that you've found the webinar today useful, and it's opened further areas of discussion and collaboration. So as I mentioned earlier at the start of the webinar, is that we'll have a follow-up of a webinar where I'll be sharing a link for a more open and informal discussion, where we'll aim to continue this conversation, where we'll have giving everyone an opportunity to share their ideas, ask any further questions, and explore any further collaborations. So please do consider looking at the new IPM registration document I've sent. We're having a wide range of industrial contacts who will be attending that symposium. And once again, I'd like to thank everyone who attended, and I appreciate taking the time out for today. So thank you, everyone, and I hope you enjoy the rest of your day. Thank you.
Thank you. Thank you.
Thank you.
You guys. Thank you. Bye.
Yeah.